Hey Super Scientists, we're looking in your molecular biology lab notebook at page 4, and we're going to be going over cell theory. On your note page, you have a little timeline that looks like this. So we'll be looking at those different scientists, and then we'll also be looking at the different components of cell theory to have a better understanding of the scientists' research and their development that led to cell theory. There were a lot of different scientists who did research on microorganisms and using cells or looking at cells under the microscope. However, there are several scientists in particular whose research led to the development of cell theory. One of those scientists is Robert Hooke. Robert Hooke did research about 1665, and his research initially was looking at cells and looking specifically at cork cells. So he was the first person who was able to see cells and to describe them. The cells that he was looking at were from a cork tree, and this is an example so you can see what a cork tree looks like. The bark is being harvested, and that is what Robert Hooke was able to look at. So if you've ever used a coaster to set a drink on top of on a table, a lot of the times the coasters will have cork on the bottom. So as Robert Hooke was looking at cork cells, he developed this little illustration, and you can see that it's got little boxes, so these tiny little openings it looks like throughout both of these little pictures here. And this is a quote from Robert Hooke. As he was looking at these cork cells, he saw these little boxes, these tiny little rooms. And so he came up with the name cells from the Latin word cellula, which just refers to rooms or little boxes, little structures. And that's what he thought that cells looked like. Another scientist who did research is Anton von Leeuwenhoek, 1673. He developed a lot of different microscopes, and some of his microscopes are still around today. So you can see that the microscope looks a lot different than the microscopes that you may be familiar with now, the compound light microscopes. Anton von Leeuwenhoek is referred to as the father of microscopy because of his development of so many different types of microscopes. And it basically used kind of like a little glass marble kind of thing as the lens to help project or blow up the images that you were looking at. He was able to see a lot of different types of microorganisms. He observed protists, blood cells, bacteria. Those are some of the big things that he's attributed with. So he did some drawings of blood cells based on what he was able to see, the red blood cells specifically. And he saw lots of different types of protists, which are either unicellular or multicellular organisms, if you're looking at something like Volvox, and he referred to these as animalcules. And the reason why he said that is because he thought they looked like animals. They were moving around, but they were really small, and we know that our stemcule refers to something that is going to be smaller, like molecule, for example. And so he drew these little illustrations of things that he was observing when looking at these different samples, and he specifically looked at pond water a lot to see different types of protists. Matthias Schleiden is another scientist. He was a German botanist. 1838 is his time frame, and he discovered that all plants are made of cells. So as he was studying different types of plants, he was able to use a microscope and lens kind of format like von Leeuwenhoek was using, and he was able to see these structures that are plant cells. His counterpart, Theodor Schwann, 1839, discovered that all animals are made of cells. So he discovered our roundish kind of animal cell structure. And an easy way to remember that Schwann discovered that animals are made of cells is that Schwann sounds like swan, which is an animal. So Schwann discovered that animals are made of cells. And the last scientist here is Rudolph Virchow. And Rudolph Virchow discovered that cells form from other cells, that cells come from other cells. Its time frame is 1855. And this is a little quote from him. Omnis cellula a cellula. So cells, omni, all cells come from other cells. So he basically discovered that the cell will divide to form two new cells through this process of mitosis. The parts of cell theory are that all living things are made of cells. All living things are composed of cells. Cells are the basic unit of structure and function in all living things, and all cells are produced from other cells. So if you have a question, for example, on a test or your EOG that says, what do cells come from? The answer, cells. Cells come from other cells. The last thing on your notes is looking at this little reflection statement. How do you think the invention of microscopes influenced the development of cell theory? So think about the information that we've looked at today. Think about the development of microscopes. You can see this microscope kind of just looks like a straight tube. And this is one you may be familiar with today, this stereo microscope that's bifocal. So think about that and write down your recordings or your information, write down your observations, write down your thoughts about how you think microscopes, micro, small, led to the development of cell theory, that all cells are made of cells, cells come from other cells, and cells are the basic unit of structure and function in all living things.